There's obviously a lot that we can talk about with soil, and I'm just basically talking about the basic principles so that you can apply them to your own operations. Somebody just asked me over lunch about perennials, and it's really important if you have perennials that you keep at least part of your grazing area, keep them in that vegetative state. So even if you say, okay, this field is going to be the one that I'm going to work on this year, I'll come in and graze them, only take half of what's available to be grazed of the green leaf I'm talking about. If you take a perennial grass plant and cut it in half, like the top half and the bottom half, it wouldn't matter even if it was a perennial, even an annual like an oat or a barley or wheat or anything, take half of the leaf area and put that into one bucket. You might do this like on, say, 20 plants, and then cut it right down to ground level, take the other half and put that in a separate bucket, send them away to a lab to have a feed analysis done on them, and you'll find that the top half has double the protein, double the energy, double the minerals and trace elements in it as the bottom half. So there is no point in you having your stock graze the bottom half. If you just take the top half, you won't have any effect on the roots. The plant will regrow really quickly, and in a growing season you will get much more production just by grazing the top half. But obviously you have to be able to manage your livestock to get them in there, get them out again, and just work on that one field. Keep it vegetative. When it regrows, get them in there and graze it again. Get them in there and graze it again. Get them in there and graze it again. Once it gets like to be tall standing feed and it's gone to seed, remember it's not building soil. But you might want some of your grasses to go to seed, so just move that area around, your priority area we call it, where you're really going to focus on building soil in that area. And also with your livestock, their animal production is going to be heaps better where they're just getting that fresh regrowth all the time. All right, so why is this liquid carbon different to what we think of as in terms of organic matter? It's different because this is an anabolic process. This is a building up process. We start with some really simple compounds that come out of plant roots, simple sugars and things, and then the microbes are going to join all the elements together to make that complex polymer. They're building up from something simple to something complex. Decomposition, which is when you have plant residues, crop residues, old grass, hay, whatever it might be, and it's a complex carbohydrate, it's usually highly lignified, it's going to go from being a complex compound, microbes are going to decompose it and turn it into something simple, and it's going to get broken down into smaller and smaller and more simple things, and eventually it becomes carbon dioxide and goes back to the atmosphere as carbon dioxide. So in the same way, we've all just had lunch, and so you're now digesting food, and you're digesting food in order to get the energy from it. The energy is actually the sun's energy that was captured in a green plant, and you either ate the plant or you ate the animal that ate the plant, and you're now going to digest that food and release the energy. And when you're breathing in air that's high in oxygen, and you're breathing out air that's high in carbon dioxide. So the carbon dioxide that you're breathing out is the final stage of the digestion process of the food that you ate. So you digested it, you turned it back into glucose, went into your bloodstream, your cells used it, the glucose, for energy, and the cells were undergoing what we call respiration. During respiration, they use oxygen, that's why you have to breathe in oxygen, and they give out carbon dioxide. So we're producing carbon dioxide all of the time, and microbes in the soil are producing carbon dioxide when they're digesting organic matter. And eventually it ends up being carbon dioxide. Doesn't stay in the soil. So if we want to build soil carbon, we have to go through the anabolic pathway, through the building up process with simple root exudates and build them up into complex polymers. Something complex like corn stalks or alfalfa hay or something like that can't turn into humus. It just cannot happen. All right, so we have those two pathways, difference between liquid carbon and organic matter. This is why the scientists get confused because they think that it's all about how much organic matter. Oh, I had a crop that yielded this many, bushels per acre, or this many tonnes per hectare or something, and therefore it should have built this much carbon. Well, it doesn't work like that. So the decomposition pathway is still going to start with dry matter, still going to start with plants, you'll still have some kind of biomass there, but it will decompose and it will form carbon dioxide. Looks great during decomposition. This is saprotrophic fungi feeding on highly lignified material in the soil. If you see all that white mycelium in the soil, that's a really good sign. These are very beneficial fungi, but they're not the kinds of fungi that feed on living plants. They feed on dead plants, and they're still it is still a very beneficial process for the soil. And we know that mulch is great stuff to have, right? But it goes back to be carbon dioxide again. So on the left-hand side, you have sand. It's got lots of organic matter in it. On the right-hand side, it just goes back to being sand when all that organic matter is decomposed. So that pathway ends in carbon dioxide. Our liquid carbon pathway produces humus, and that's the one that we want to focus on for really rapid soil building. But both pathways are important. So what you're seeing there in that diagram is a green plant with, if you follow it down from through the centre, you'll see that there's exudates there, root exudates. That's the primary pathway for soil building down to our stable carbon pool at the bottom. But there over on the your left-hand side, you'll see there's a leaf litter that's composed of complex carbon compounds like lignin and cellulose that gets broken down by extracellular enzymes from things like fungi in the soil, and those enzymes actually feed into the humification process as well. So having organic matter there and having organic matter decompose is an important part, and when you think of a natural system, obviously it's going to have organic matter in it too. But we know that root inputs actually build carbon 5 to 30 times faster than organic matter. So this is the pathway that we want to look at. Now, if we're going to send soil off to a lab to have a test, as I said, most of the soil test results are useless. Your nitrogen test means nothing, your phosphorus test means nothing, but your carbon level is very important. And the lab can't measure organic matter, they can only measure carbon. So they'll probably use a combustion method. In other words, they're just going to weigh the soil, then heat it up to a really high temperature and then weigh it again. And what's been lost as carbon dioxide, they will calculate how much carbon it had in it. And it does tell you a lot about soil health. So you want to know over time whether your carbon levels are increasing or whether they're decreasing. And the reason that you want to know how much carbon you have in your soil is because it's related to so many other factors. It is the key determinant of the water holding capacity of your soil, and that doesn't matter if you're in a wet area, your soil is going to be less soggy, if I can use that technical term. If it's got high water holding capacity, it will be better structured and it won't be so subject to like treading and things by animals or vehicles. And in dry areas, obviously, we want to have high water holding capacity. That's pretty obvious to maintain green for longer when it's dry. It's also the key driver for the nutritional status of plants. I'll talk about that in a little while when I talk about microbes, and therefore for your animals and for the people consuming that food. So it's a very, very important thing to know how much of it we have in soil. And for those reasons, it's the key driver for farm profit. Unfortunately, organic carbon levels now around the world are 50 to 80% of their original level, not just here in the United States, but also, as I mentioned, places like Australia, South Africa, South America. 30% of the world's cropland has been abandoned in the last 40 years due to soil decline, and it's still being abandoned at the rate of 25 million acres a year. So why is this happening worldwide? Why is so much land deteriorating and continue to deteriorating? Why do you think that might be? I mean, why is all that soil all around the world going backwards and continuing to go backwards? We're still losing valuable farmland. It's being totally abandoned. It's not productive anymore. We've done it, but what did we do? Because we have to stop doing whatever it was that we did, right? Yeah, insecticides are kind of a symptom. Like you wouldn't apply an insecticide unless you had an insect problem. So why did you have an insect problem? Why did you have a fungal problem? Why have we got all these problems? We've killed the soil, and how did we kill it? We didn't actually kill it with the insecticides and fungicides. We only use them now to sort of try and solve the problem. We're dealing with symptoms now. How did we kill the soil? We deprived it of carbon. We took all the plants away. Exactly. It's so simple, isn't it? And so theoretically we should be able to put them back. So if we look at the statistics, they actually show that 50% of the world's cropland is bare in any 12-month period. And any of you who fly around either across the United States or if you fly anywhere else in the world, just look out the window and look down on agricultural land and you'll see how much bare ground there is. As I said in Portland yesterday, high rainfall, mild temperatures, about 70% of the cropland that I was looking down on was bare. And the soil is dying because it's bare. And that's why we're losing it. So it doesn't matter whether it's bare because we cultivated it or whether it was bare because we sprayed it out. Like, where is the photosynthesis there? Where are the green plants? We adopted no-till. A very large percentage of Australian farmers adopted no-till back in the 70s. It was very, very rapidly taken on. The government promoted it very highly and encouraged it, and lots of information for people for like swapping over to no-till machinery, particularly like disc openers and things. And our soil carbon is still going down. It never ever increased. It never made any improvement to our soils at all going to no-till because we never went to cover crops. Where is the green? There is no difference between really doing that and doing that in terms of looking at it from the perspective of the soil. So how can we know what we know now about green plants to actually build topsoil? Well, it doesn't matter what it is that you are using your soil for, whether you're growing a sugar beet or alfalfa or corn or potatoes. I have to say potatoes, don't I? I haven't even got potatoes on the list. My goodness, how could I have left them off? I've got vegetables. That'll do. All right. It doesn't matter what it is that you're producing. You are actually just farming light. You are a light farmer, so you have to think about photosynthesis. And there's two things about photosynthesis that we need to think about. Not that hard. We have to think about photosynthetic capacity and we have to think about photosynthetic rate. They're very different and they're both things that we have to consider. So what do you think photosynthetic capacity might mean? Let's just say this room that we're in now is a field. How would I know, just looking at this room, if this was a field, how would I know how much photosynthetic capacity I had? What would I be looking at? What would I be looking for? How much green I have. And if I was looking to see how much green I had, what important things would I be looking at there? Like, surface area, like how much of it is covered. I mean, there could just be a few little clumps of green stuff and lots of bare ground in between. So if it was a perennial pasture, for example, I'd want to know how much ground cover I had. Or even, that's going to be an important thing, isn't it? Like how much ground is covered. And then I'd want to know, well, how tall were the plants? And did I have lots of different kinds of leaves there? Lots of different shaped leaves. So this is one of the things about our multi-species covers, is that we're intercepting a whole lot more light. In fact, grasses don't really intercept that much light. They're not that good. Because when you look, like, they're pretty thin leaves and the angle that they sit at, something like a sunflower or some of your forage brassicas and things with the nice big leaves are actually collecting a whole lot more sunlight. And if we've got lots of different shaped leaves of different angles and different kinds of plants in there, and also it would be great to have things, obviously the original prairies had plants that grew at different times of the year, so whenever it rains there's a chance that something is going to be able to grow. So our photosynthetic capacity is how much green, and there's lots of things that factor into that. How much photosynthetic capacity is there? Well, we know that there's zero, and in Australia when we went to no-till, we still got no photosynthetic capacity. So we've got things like, your cover crops, multi-species covers that you can put in, or in your vegetable garden, make sure that you've got all the ground covered with lots and lots of different vegetables all in together, and put lots of flowers in there so there's no spaces for weeds. That's a photo from Gabe Brown. That's his chaos garden where he put 20 vegetables and 20 flowers, threw them in the seed drill and just planted them all together, and had a very bountiful crop with no need to use any chemicals for anything. This is what we call pasture cropping in the United States. In Australia, where we grow cereals into perennial grasses, so we've always got ground cover. And an example from South Africa in a very, very dry area in a citrus orchard where they've put a plant that's in the succulent family. It's got big pink daisy flowers. It's a kind of a portulaca, but I think you use that word portulaca to mean other things here in this country, so I'll just say it's a plant that's in the daisy family, but it's very, very drought tolerant, and they've just planted that down the rows. It's made a huge difference to the health of their citrus trees, putting those other plants in there rather than having bare ground that they kept bare with Roundup, glyphosate. So just to show an example from Western Australia where just putting some plants in where you can see what it's actually done to the sand. The farmer who owns these cattle actually is here in the United States right at the moment, Grant Bain. He's at there's a Cattlemen's Association meeting happening in San Antonio in Texas starting tomorrow, and he's just come over for that. So it's interesting that he's here in Australia at this time. But he had thousands of hectares of sandplain country that was originally perennial grasses at the time of European settlement, but because of the hot, dry climate in that part of the world, and having animals in contact, not rotationally grazing animals, they've lost the ground cover. So, and there's no seed bank anymore because a lot of the sand is blown. So a lot of farmers in that area, and Grant was actually the pioneer of this, formed a group called Evergreen, and they planted perennial grasses back into that sandplain. And it's very interesting to see, if you go out with a spade and dig underneath those grasses, and have a look and see what they've actually done to the soil, what you see is that they don't look all that flash. And it's like over 100 degrees for probably 5 months every summer, and it doesn't rain in summer. So those grasses have been grazed 3 times. So that's been grazed 3 times and it hasn't rained. So it's doing extremely well. But when you look at the sand underneath it, you can see how it's really changed the colour of the sand. And it's actually building topsoil. So it's not just feeding animals, it's stopping the sand from blowing, and it's building topsoil. So if we were to take a soil core, like use a metal soil coring device and take a soil core down through the centres of those plants, and then take soil cores in the interrow where there aren't any plants, and have a look to see what the plant has actually done to that soil in terms of its nutrient status. This data was collected by the West Australian Department of Agriculture. And what they found was that first column where it says between, that's where there's no plant. That's between the rows, the interrow. And where it says within is where a soil core was actually taken in that row that's got plants growing in it. And you can see that the organic carbon, even though it's very low, remember I said that most Australian soils we now have less than 1% organic carbon, but where the plant is, there's 4 times as much. That's a lot. That makes a huge difference to things like how fast water can infiltrate and how much water can be held. And how much life there is going to be under those plants. You can see that phosphorus availability has tripled, and potassium availability has tripled, and sulphur availability has almost tripled, and the pH has gone from 5.8 to 7.1. In other words, it's normalised. So the plant has built soil underneath it. How many of you have heard people say that plants take from the soil? It's a really common saying, and they'll tell you this at universities. They still tell you this at some universities, I should say, not all. Plants take from the soil. And what do they take and where do they take it to? Whenever people talk about plants taking stuff from the soil, I always have this vision of them getting up in the middle of the night and running off and taking stuff somewhere. Like, a plant can't possibly take from the soil. A plant is the only thing that can build soil. And there is so much evidence for that. I don't know where that plants take from the soil saying ever came from. So what about photosynthetic rate? So that's our photosynthetic capacity, is how much green do we have there, how much of the year is it green, how many different shapes of leaves have we got. So let's just say now that I've got 2 pots here in a glass house and they've got wheat in them, and it's the same variety of wheat, and I planted at the same time, and the soil is exactly the same, the moisture content is exactly the same. Do you think that there is some way I could vary the photosynthetic rate? Do you think that we could have wheat in one of those pots photosynthesising at a different rate to the other one? And how would I know? Yes, you could take away the light. Well, we're assuming that we're going to have the light, the water, all of those physical things are going to be the same. How would I know how fast the plants were photosynthesising? How could I measure it? Could I put a number on it? Brix, who said that? Right, so I'm going to use a little device called a refractometer that's going to measure the refractive index of the sap of the plant. You should all have one. I noticed that they've got them on Amazon now for about $21. When they first came out, they were about $200 and then $100 and then $80, and I'm not sure what the quality of the $21 ones is, but it's going to be better than not having one at all. You also need to have a garlic press, a stainless steel garlic press, so that you can squeeze some sap out of your plants, and you take a sample of sap, you're going to put it on a little glass plate on your refractometer, you're going to hold it up to the light, and you're looking at the refractive index. You're looking to see how much dissolved solids are in the sap of your plant. If you put distilled water on there, it's going to measure 0, so it's got nothing in it. You want the sap of your plants to be absolutely loaded with sugars and minerals and vitamins and trace elements and all kinds of things. You want it to have a really high refractive index, because then it's going to taste great to animals, or if you're producing something like grain or potatoes or whatever it might be, it's going to be nutrient dense. So get a refractometer, start measuring Brix, look it up on the internet, download the Brix tables, figure out where you lie in terms of is this good, bad or intermediate, because every kind of plant's going to have a different optimum level, and also it depends on whether it's a sunny day or not, because you're measuring photosynthesis. But let's just say, okay, so I have a refractometer, I'm going to measure the Brix levels of these plants. Is there some way that I could apply something to one of these plants that would actually make the Brix level go down compared to the other one? Do you think that it's photosynthesising, it's in the light and it's got plenty of water? It's a good point. If you put it in the dark, it's going to stop photosynthesising, which is another good point. If you're going to measure Brix, you don't go out and do it in the middle of the night, right? So you would actually probably pick to do it at 2 or 3 o'clock in the afternoon, and you'd be consistent about that, because if you did it at 2 o'clock one day and then at 9 o'clock in the morning the next day, you're going to get different answers. So start keeping records of your Brix measurements and figure out for yourself how photosynthesis affects it. So Brix is going to start off low in the morning and increase during the day, and then at night it's going to drop when the plant's going to download all its sugars down into the soil to feed the microbes. And then the next morning it's going to start low and it's going to increase again. So I've got 2 plants here, the light's the same, the time of the day is the same. I could put something on one of those plants and reduce its photosynthetic rate. Not glyphosate. Water-soluble nitrogen. Why would putting water-soluble nitrogen on a plant make the Brix level go down, do you think? Makes it lazy, absolutely. Why does it? It doesn't have to photosynthesise to feed soil microbes anymore. So its Brix level will go down. Doesn't have to work, exactly. You just put it on welfare. What could I put on a plant, do you think, that might increase its photosynthetic rate? You could put compost tea on it, exactly. And why do you think that might increase its photosynthetic rate? Sorry? Well, no, it's just you're actually making the plant aware that it's in a microbially rich environment and it needs to feed microbes to get the nutrients that it needs, so you're just stimulating it. It's called a biostimulant, so something that stimulates a plant. A plant will produce a lot more exudates from its roots when it's in a microbially rich environment than it will if it's just in dead soil. So photosynthetic rate is something that we can increase by a factor of 10 or 20. So photosynthetic capacity, sure, you can fill the whole field with lots and lots of plants and have great ground cover and lots of different kinds of leaves, but you have to think about how fast they're photosynthesising too, because then you can come in on top of your photosynthetic capacity and hugely increase the rate. And that's where we see people making really dramatic increases in soil building. Because, for example, if you planted a multi-species cover, you could have 20 different kinds of plants in there and all different functional groups, and you go to all that trouble and then you put fertiliser under the seed or you use seed that had insecticide or fungicide on it, you've really shot yourself in the foot in terms of photosynthetic rate. And if you're using a refractometer, you'll soon find that out, and you'll also notice that you're not building anywhere near as much soil as you could. So the liquid carbon pathway actually functions in two directions. We have carbon moving from plants into the soil at the same time as we have the nutrients moving from the soil into the plants and therefore into the food chain, like into your animals and into people consuming that food. But what you're measuring with your refractometer is how fast that's happening. So when you're measuring Brix, you're not only measuring how much sugar is in your plants, but you're measuring how much nutrient is in your plant, and that's why it's very important. Okay, so the question was, I don't know anything about that product, but there was a product called Brix Mix that you could give to plants. And the question was, would that have the same effect as using nitrogen or phosphorus? The answer is no, it wouldn't have the same effect because there are only two things that shut down photosynthetic rate, and that is nitrogen and phosphorus. So all of the other things like, say, lime or gypsum or any like compost extracts or milk or any kind of biostimulant or anything else, really there is nothing that shuts down photosynthesis other than nitrogen and phosphorus, other than poisons, obviously. Glyphosate does a pretty good job. But yeah, so that's a good question because they're the only two nutrients that do affect photosynthesis. So we've just got our plant, and the green, the yellow is the roots, and then the little blue spots are just all the microbes living around the roots. Again, that's an NRCS diagram. But those plant symbionts really increase photosynthetic rate. So the more things like trichoderma and mycorrhizal fungi we have around plant roots, the faster they will photosynthesize because they're feeding those guys. And they're feeding them because they're very protective of the plant, and they're also bringing lots and lots of nutrients to the plant. So they increase the mineral and the content, mineral and forage content. So we've just talked about using a refractometer. I just wanted to show you this graph here. This is from Alan Williams. Some of you may know if you've anyone here attended the Soil Health Academy with Gabe and Alan and Ray. I know you had Gabe here last year. Anyway, Alan Williams works with Gabe and Ray, and he has a PhD in animal nutrition, and I think he may have a PhD in something else as well. Anyway, it's a very clever man. So this is his data, and what you're seeing here is that along the bottom axis is Brix level. So Brix is measured as a percentage of dissolved solids in the sap. Most of the refractometers that you have will go up to 30, and just so make sure if you're buying one that you get one that measures up to 30, because some of them only measure up to 10 and they're just for people making home brews and things like that. So make sure you get one that goes up to 30. We have farmers in Australia with Brix levels up to 30 in their cereals. Ian and Diane Haggerty, who just spoke at No Till on the Plains last week, their wheat Brix is up to 30. I've actually seen it. I've been in their field when they've been measuring it. So it's even off the scale here. But what this shows you is that on the vertical axis, we've got average daily gain in pounds per animal unit per day of cattle. And you can see that the average daily gain per day is like almost a straight line relationship between that and Brix. So what that means is that for the same weight of forage, They could be consuming the same dry matter of forage, but if it's got a higher Brix level, they will gain more weight from that same dry matter. Get that? Because it's got more energy, protein, minerals, nutrients, vitamins, enzymes, like everything that animals need if it's got a higher Brix level. So it's really important. So what did I say about forage before? The top half of the plant has double the protein, double the energy, double everything. And you want to keep plants in that vegetative state, because not only are you going to get higher dry animal live weight gains, but you're going to be building more soil. You can have your cake and eat it too. It's really, really important to get that grazing management right. And if you can't manage it over a large area, just concentrate on some smaller areas and see the effect of doing it that way. So building topsoil is a biological process, as well as obviously feeding animals. And of all of the microbes that are important, mycorrhiza are really key, or we used to think that they were. I think now the recent work on endophytes is showing that there's a lot of things that may be even more important than mycorrhiza that we didn't know about, and I am going to talk about endophytes very briefly as well. Again, an NRCS diagram just showing you the leaves, the roots, and then the golden threads are basically the mycorrhiza. They hugely increase the surface area of plant roots. This one is an actual photograph, so that's a plant root tip, and all the things that look like little hairs or like a bottle brush or something coming out of that plant root tip are the hyphae of mycorrhizal fungi. So these is a fungus that's living in a symbiotic relationship with that plant in the soil, and you can see how hugely it increases the surface area of that plant root. But it's not just increasing the surface area for absorbing things out of the soil. There's going to be all colonies of bacteria all around those fungal hyphae that are getting carbon from the plant root via those fungal hyphae, and then so it's feeding a huge area of soil as well as extracting nutrients from a huge area of soil. It's keeping a lot of other microbes alive as well. And remember the diagram that I showed this morning? If you think of that, if a plant root tip is like that with all the fungal hyphae sticking around it, you can see how when they pull the soil particles together that they can actually form those aggregates. So we need to make sure that we have roots colonised by mycorrhiza to form these aggregates in the soil. And if we look inside a plant root, this is a cross-section looking under a microscope, those little things that look like little trees or broccoli plants or something, this is a close-up of one, or like an animal lung. This is the arbuscle, so they're arbuscular mycorrhiza. And this is the exchange site. It is like an animal's lung. It is like our lung even, where we're exchanging oxygen and carbon dioxide. But this exchange site within the plant root is where we have sugars and things that the plant is manufacturing in photosynthesis are going into that thing that looks like a lung, into that arbuscle, going one way, and then the other way we have all the nutrients and the water and everything that the mycorrhiza is bringing from the soil going out into the plant. So the mycorrhiza is feeding the plant at the same time as the plant is feeding the mycorrhiza. Now, I asked before what would happen, and this increases the photosynthetic rate of the plant. If it's got all these guys inside the roots, when you stain the roots and you can see all these fungal filaments in the root, it's got to photosynthesize a whole lot faster to feed all these guys. It gets benefit from it, that's why it does it. But if we come along and apply some water-soluble nitrogen to that plant or put some water-soluble phosphorus on that plant, it doesn't need the mycorrhiza to bring the nitrogen, doesn't need mycorrhiza to bring the phosphorus. It closes those arbuscles down. And you can actually see, if you're looking under a microscope, you will just see that that arbuscle will just close like that. Within 2 minutes, it'll be completely gone. The plant shuts them down. So when the plant shuts them down, it's not channeling carbon to the soil anymore. It's not building soil anymore. And its photosynthetic rate drops. That's why you can pick it up on a refractometer. Half an hour later, after you've applied the N, the Brix level will be lower. So you've really, really shot yourself in the foot by doing that. That's a good question. Off the top of my head, I can't say. I would probably guess at 500 or something, but I'm only guessing. I could find out for you. So this is a photograph showing the fungal hyphae coming out into the soil, carrying carbon out. If you actually want to stain plant roots and look at mycorrhiza, there's lots of information on the internet as to how you can do it yourself. You can do it in the kitchen, at home, using an aniline dye. I mean, it's quite a complicated process, but some people like to do it because they actually like to stain the roots to see whether it's a blue dye that you use to stain them with. And you'll find information about how to do it and what magnification to use and everything. Off the top of my head, I'm not sure. And this is a very famous photograph that lots of people use, but the point about this one is that this is a little pine tree, little pine seedling. So we've, got the roots up. Sorry. Oh, I don't think I've got a laser. I don't know whether I've got a laser on here. Anyway, you've got your leaves, and then the roots are the yellow bit underneath that are just about the same size as the leaves. The rest of everything that's in that photo is the mycelium of mycorrhiza. You can just see how a little seedling like that getting established, how much difference that makes to how much soil it can explore and how biologically active that soil is going to be if it's connected to mycorrhiza. And again, if we were to annihilate that, like a fungicide is probably the worst thing you can ever use in your crops or pastures. And some people apply fungicide because it doesn't cost that much more just to throw fungicide in with anything else that they're putting out. Oh, well, it'll only cost a few dollars more to put fungicide out, we'll do it. And just incredible damage to your soils using a fungicide. You can see that the root is just the bit going down the centre of that photograph, and all the rest of that are hyphae of mycorrhiza. And these plants are connected with mycorrhizal fungi. So it is the highway and the internet of the soil. They're transferring nutrients between each other. They're sending messages to each other. So if a plant is attacked by something like, I don't know what kind of pests you have in this part of the world, but some leaf-chewing or sap-sucking insect or something like a aphid or something like that, it will actually send messages to other plants. Like the aphids are coming, and that message goes right through the mycorrhizal network, and plants that haven't been attacked yet will protect themselves from insect damage. But of course, if we're using fungicides, then we end up with more insect damage by using fungicides, strange as it may seem. So these little guys are the highway and the internet of the soil. They're inhibited by inorganic N and inorganic P. So how do plants actually respond? If you were looking to see what would happen when you add inorganic nitrogen to the soil, well, this is a pretty classic sort of a photograph. You'll see this many times in many soils around the world with seeds that are treated, got fungicide and insecticide on them. You'll notice that the roots coming off those seeds are very white and very clean. They don't have any root hairs. They don't have any soil sticking to them. So basically, those plants are completely unprotected, and even though they're treated seed, they're going to be very vulnerable to attack by insects and pathogens because they haven't got their microbial partners helping them. And you can see that the soil's got no structure as well. So unfortunately, this is how we've got to where we are in agriculture with all the chemicals that we use. The plant on your right has had 100 pounds per acre of monoammonium phosphate under the seed, very short root system. The roots haven't got much soil sticking to them. The two plants on your left grown in a biological system. The wheat is the same age, deeper roots, more soil forming going on around the roots, more biologically active around the roots. So this is a farm in Norway. It was an organic farm. They were trialling 20 different kinds of compost. They made compost from things like pig slurry, chicken litter and forest timber byproducts and all sorts of things, 20 different kinds of compost. And we were digging the plants out and having a look to the roots to see if we could decide which compost was the best. And every single lot of plants that we dug out, we noticed that they had, you can't see it so much when they're all together there, but if you tease a couple of plants out by themselves, the seeds are about halfway down in that photograph. I don't know whether you can pick the seeds out. You'll notice that the roots coming out from the seeds, the seminal roots, the first roots that the plant produced are clean. We didn't shake the soil off or anything. There is no soil sticking to those roots. The roots that are coming out of the crown of the plant, so what happens is the seed germinates, it sends some roots down, it sends a coleoptile up, which is the shoot that goes up to the soil surface. Then when it hits the soil surface and starts producing leaves, then there'll be some roots will come out from the crown. The crown roots all had lovely rhizosheaths on them. There's another photo there. Can you see? I haven't got a pointer on here, I don't think, but can you see the seed? And everything underneath the seed is clean, which is not good, not healthy. Okay, thanks, Marla. All right, so the seed is there. Okay, can you see the seed there? There, okay. So see all the roots coming down from the seed are clean. That's not good. The roots above the seed have got lovely rhizosheaths on them. So what's going on? This is an organic farm. They're trialling all these different composts, and I'm just like, well, I have no idea. I couldn't work it out. I said, it looks like there's some kind of toxin in the soil. Maybe whoever owned it before put massive amounts of insecticide or something like that. Anyway, we went back and we had lunch in the farmhouse, and then I said to Sarah, go backwards, the lady that's looking at me there, I said, you wouldn't have put any nitrogen in this trial, would you? I mean, you know, you want it, you're transitioning to organic, you're trialling compost. It's the only thing I could possibly think of that could do that. And she goes, oh, yes. She said, the scientist at the university, I think it was the professor at the university who designed the trial for us said, because all these different kinds of composts had different carbon to nitrogen ratios, so things like pig slurry obviously had a lot more nitrogen in it than something that was from the paper pulp industry, that to take out the difference in carbon to nitrogen ratio, she said, we put 80 units of N under the whole trial. And I said, you put it under the seed, didn't you? I said, the machine that you planted with put nitrogen in under the seed. She goes, yep. I said, right, well, that explains exactly what we're looking at. So what you're seeing there is the first time I've ever seen the two things on the one plant. You're seeing clean roots where the nitrogen went, and you're seeing rhizosheaths on the roots where they were in contact with the compost above the seed. So the other thing about rhizosheaths is that a farmer from Washington State just sent me some photos the other day, and it was cereal rye that he was growing as a cover crop. He said, I can't understand it, Chris, I've got really excited about rhizosheaths now, and I'm going out and I'm digging holes. He said, in one field we've got really great rhizosheaths on the cereal rye, in the other field the roots are all bare. I can't work it out. I said, what can you tell me about the seed? He said, the one with the rhizosheaths was organic seed, so it didn't have any chemicals on it. So the rhizosheaths are an indicator that the plant is forming a relationship with microbes. That's what it's telling you. And if you're not getting the microbes there, if you're not getting the rhizosheaths there, you need to look at, well, the other seed that wasn't organic, I don't know how it had been treated or how it had been grown, but it obviously was antimicrobial. And we know there's off-site impacts of high analysis fertilisers. I mean, I don't need to tell you here in the United States that 49 states have some serious level, actually, of nitrate contamination in the water, the groundwater, and it's a potent carcinogen at 2 parts per million. I think EPA standard is something like 10 parts per million or 11 parts per million. It's carcinogenic at 2. So that's something that we seriously need to consider, as well as what it's doing to our soils. It's what it's doing to our water. So we've tried to replace the biological activity that we should see around plants with chemical fertilisers. We now know that plants supported by high analysis fertilisers actually can't get the minerals and trace elements that they need, which is one reason why their Brix levels are so low. And we know that there's like soil degradation issues associated with using those things. So how can we support microbes rather than using high analysis fertilisers? So if we're just going to switch over to another presentation here. Oh, thanks, Hayley. That was quick. Right. So a recent census of life showed that there was 550 gigatons of carbon-based life forms on the planet. Don't ask me how they worked it out, but somebody did. And of those 550 gigatons of life, 450 gigatons was in the form of plants, which is hardly surprising because in most regions where you go, most of the living things that you see actually are plants. What was interesting is what makes up the other 100 gigatons. So most of the life on the planet is plants. What makes up the rest of it? Well, it's things we can't see. 93% of the rest of the life on the planet are things that we would need a microscope to see. The protists, the archaea, the fungi, the bacteria comprise most of the life on this planet apart from plants. And if we look at that diagrammatically, you see the archaea up there in the orange, the protists in the orange, archaea purple, fungi green, and bacteria whopping 70% of that. So these little guys and gals are basically making up most of the weight of life on Earth. And things that we can see when you think about life and living things, you probably think about animals and birds and fish and insects and earthworms, all those kinds of things, and people. When you add all of that stuff together that we normally think of as being life on Earth, it adds to 7%. And in fact, humans are only a tiny, tiny fraction of that. We are 0.01% of the biomass of life on Earth. And that's by weight, talking by weight. When we look at the numbers, what we find is that are even more staggering, because 1 teaspoon of healthy soil, and I'm talking about nice healthy soil from around the rhiza sheaths of healthy plants, has got more microbes than all the humans on Earth. So even a couple of grams or a couple of, oh gosh, I don't know what that is. Grams is not very much. Even a half a teaspoon of really healthy soil from around the Rhizosphere has trillions of microbes in it. We've got something like 7 billion people. So we're talking trillions, another order of magnitude of life just in a half a teaspoon of soil. And then if we look in the rumen, like the rumen of a cow or a sheep or a goat or a ruminant animal that has this big vat where it's fermenting cellulose, it's an incredibly rich microbial environment, and 1 drop of rumen fluid contains 10,000 times more microbes than there are humans on the planet. 1 drop of rumen fluid. So it's really hard to get your head around these kinds of numbers, right? But even on a weight basis, even if we're weighing them, microbes don't weigh much. Even on a weight basis, we don't figure in the biomass of life on Earth. We're 0.01%. We've done a lot of damage, haven't we? 0.01%, but we can do a lot of good too. And now any of you who are interested in human health will know that we, on a cell count basis, in other words, if you count how many microbes live in us and on us, that we have about 1 trillion human cells and about 10 trillion bacterial cells. So we are 10% human on a cell count basis. And I think there was a book written called 10% Human. I haven't read it, but some of you may have. And then if we look at our genes, like what really runs as our DNA, we find that we have about 300,000 human genes and about 100 times more that in bacterial genes. So we're maybe 1% human. Now think about that. We think of ourselves, right, as being, well, human, I guess, but we are microbe taxis. We are absolutely covered and full of microbes, and they have a huge impact on our health. If we want to be healthy, we have to look after our gut microbiome. Your gut microbiome is running you whether you want to believe it or not. And almost any magazine that you pick up on human health today will talk about the gut microbiome, and of course the health stores will all be trying to sell you something, kombucha or whatever, that's going to boost your gut microbiome. But actually you just have to be really careful about what you eat, because that's what's going to determine what goes on in your intestine. And we know now that our gut microbiome is running our brain function as well, the functioning of your brain, running how you feel, what your mood's like, probably linked to a whole lot of mental health issues. And I can't help wondering when you look at the standard American diet, the SAD diet, and the huge increase in mental health issues, homelessness, all these kinds of things that seem to be linked to mental issues, that is it got something to do with what people are eating. The other day I did a workshop in Portland, Oregon, and a couple of the women had brought their babies along, and they were women who were, growing organic food and being very conscious of what they fed their kids. These babies were absolutely gorgeous, bright, quiet, like just beautiful children. They didn't have any kind of instability issues or any mental issues. And I think that, you know, we have to look really seriously at the relationship between our gut microbiome and our brain function. But that wasn't what I'm here to tell you about today. What I wanted to talk about was what happens with the soil microbiome and how that determines how our plants and our animals function. So the situation with plants is similar to the situation in humans. There are more microbial cells in and on your plants than there are plant cells. Your plant is just a microbe taxi as well. And now that we know more that we know about soil, we know that plants are actually cultivating microbes, farming microbes, using microbes for their advantage. They're probably a bit smarter than we are in terms of how they actually utilise the soil microbiome. So more microbial cells within plants than there are plant cells. So all plants and animals are embedded in a microbial world, and we have a microbial world embedded within us, and we need to get used to that idea pretty quick, because it's actually a good thing, because microbes are capable of performing all sorts of amazing tasks that we can't perform. We actually need to have them, and we need to look at how they work. So they're very tiny. They can't see, they can't speak, they can't hear. So how do they communicate? How are they so extraordinarily capable of, when you think about it, these little dumb critters that can't talk to each other, how did they get their act together to join all those elements together, the carbon, the hydrogen, the oxygen, the nitrogen and everything, and make humic polymers? How do they do that when they can't even talk to each other? How are they communicating, do you think? Yes, they are communicating non-verbally. Correct. The lady down the front says chemically, biochemically. Yeah, quorum sensing. How would, if they were communicating using quorum sensing, what kind of signalling molecules are they using? Quorum sensing is actually how they coordinate their activities, but they're sending out some kind of a biochemical signal, aren't they? Like, because how does our body work? You've got heart, liver, lungs, spleen, adrenals, hypothalamus, a whole lot of different organs in your body, right, that are all working together so you function as a unit. Right? Every organ in your body is doing something, but you're not thinking about that right at this minute. Your heart's beating, your lungs are breathing, your liver's detoxifying your blood. Yep. That's good. That's good. That's very good. That's very good. So if your body, let's say there might be something that you did need to eat, you might actually get a craving for something that had some vitamins and minerals in it that you may not necessarily find in that sugar-coated donut, that you might get a craving for eating a nice apple or something like that to get but anyway, but the point being that your body is coordinated. Your hypothalamus, your pituitary, your adrenals are telling all of the other organs in your body all of the time what to do, and they're sending out signals, and those signals are in your bloodstream. So for example, if your pituitary wants to send a signal to your thyroid, which is here in your throat, to get moving, do something, we need some more T3 and T4 being produced. It will send thyroid stimulating hormone, TSH. It will, your pituitary's up in your brain. It's going to make TSH. It's going to go into the bloodstream. It's going to circulate in your whole body. And you know what? Your liver and your kidneys and your spleen and everything else is going to totally ignore it because it's not meant for them. It's just meant for the thyroid. So the thyroid has a receptor for TSH. When TSH is elevated and it's in the bloodstream, the thyroid goes, oh, pituitary's telling me I've got to get moving, and it will respond to that. So that's how our body works. There's a whole lot of signals there that we ignore because they're not meant for that organ, and there are specific signals that are meant for specific organs. That's how the soil works too. So the microbes are all communicating with each other. Look, we have to join these carbon and nitrogen and hydrogen and oxygen level atoms together to make this humus. But why do they do that anyway? Why do they go to all that trouble to coordinate their activities and make humus? They could just use that carbon and have a big party. They could just use it for energy. They could go and have a lot of fun, reproduce and everything, you know, using all that energy, couldn't they? But instead of doing that, they've actually gone to work and they've made polymers out of carbon and hydrogen, oxygen, nitrogen. Why did they do that? To modify their environment. Did they make it a better environment or a worse environment? Yeah, presumably better, because they've made an environment that now holds more water and it's got better structure. And what do you think are the advantages, if there's an advantage of having an environment that holds more water and it's got better structure and more nutrients are available? And why would they want to do that? Sorry? Exactly. The plant will grow better. So the plant will photosynthesize more now, which will feed them. So they can reproduce and have more of them because now the plant is channeling more carbon into the soil to feed them all. So the more soil they build and the more chance that the plant is actually going to survive in a dry time, or is going to survive in a really cold time, or is going to survive if there's insects around or fungal pathogens around, because it can defend itself now. It's got a really good immune system. So they are building a more productive, resilient plant that is going to feed them. So that's why they go to all the trouble to make humus and build soil, because why would microbes bother doing that? There's got to be an advantage in it. So they communicate extremely well, and they're talking to the plant all the time, and the plant is talking to them all the time. And we're just starting to pick up on some of these signalling molecules and seeing how they work. So like I said, that just tells you it's like communication in the human body. There's biochemical signalling going on in your body all of the time. So the plant holobiome is the plant and all the microbes that are associated with it. It is everything above ground, everything below ground, all the microbes on it, around it and in it. And the plant holobiome is made of the phyllosphere, the rhizosphere and the endosphere. So we hear a lot about the rhizosphere because that's something we can dig a plant up, we can look at it, we can see what's going on. And in the past there's been a lot of research into things like mycorrhizal fungi that obviously live in the rhizosphere. We're now starting to look more at the above ground part as well, the phyllosphere, which is basically everything that's above ground. There's a lot of signalling goes on above ground. And one of the things is that if you were a plant, that's what it would look like to you. Like there's a whole lot of molecules out there that mean stuff that we can't see them. Sometimes we can smell them if it's an aromatic plant. If you touch something like a mint plant or something like basil, you can smell it. But for the most time, we can't detect all these chemicals. But other plants can. So plants know what other plants are growing near them because of all the chemicals that are in the air, and insects know. So an insect, for example, this is a classic example here. If you measured the Brix of that lower leaf, it's much lower than the Brix of the higher leaf. The more newly emerged leaf in this case has a higher Brix level. The insect is picking up on low Brix. So we know that insects will not attack leaves of Brix 12 and above. And there's lots of information on that. So if you're finding that your plants are Brixing at 2 or 3 and you're needing to use insecticide, that's why you're needing to use insecticide. If you can get the Brix level up, you'll see that insects don't attack the plants. So this example of what it's actually like for plants, all the signals that they're picking up, it would be the same that this room at the moment is full of all kinds of whatever you want to call them, electromagnetic signals, I suppose. If you've got a phone, it can pick up an SMS or you can get a call on it. If you had a television receiver, you could watch something on TV. If you had a radio, you could listen to something on the radio. So this room is full of all those signals, and if you have a device that can pick up one of those signals, then you can read it. You can read the signal, right? The plants are reading these signals all the time. It's just that we can't pick them up, but insects can. So then the rhizosphere, obviously that's the bit around the roots, and we're all very familiar, I think, or most of us in this room, with what happens around the rhizosphere. We want to look for rhizosheaths. I talked about, that all the life that happens inside there. The fact that you won't get rhizosheaths if you use nitrogen. So the plant on the left, that's just across the fence. That's oats of the same age. The soil's collected on the same day. With nitrogen on the one side of the fence, without nitrogen on the other, you can't even see the roots. There's so much soil around them. But when we stand on the top of soil, we're actually standing on the top of this other world in terms of rhizospheres, all the different root architectures, and the fact that we know that the plants are actually joined together with common mycorrhizal networks. So all of those plants are going to be linked underground. They're going to exchange nutrients and water and those kinds of things. And the most well-known symbionts in the rhizosphere are mycorrhizal fungi, and they form common mycorrhizal networks. So I mentioned about plants linking up underground. There's been a lot of research on that as to who's putting carbon into the system, who's using nitrogen, who's using phosphorus, etc. And things like looking to see what happens when you put different kinds of plants together, because plants with different functional groups will sometimes work better together than others will. So there's a matter of looking at a whole lot of different combinations, and this one's just looking at monoculture sorghum compared to monoculture flax. And when you put sorghum and flax together, you find that the flax increases in it biomass by about 300%. The sorghum increases a little bit. So a lot of research now into what kinds of things can we put together and which plants will increase the most by putting them together. So if you have a look in your green cover seeds soil health resource guides that you have in front of you, there'll be a lot of information in there on relay cropping, polycropping, interseeding, companion cropping, whatever. All kinds of different ways of putting plants together. And that's going to be the future of agriculture, I have no doubt about that. We are going to move right away from monocropping because for a start, it's very expensive because you have to put so many inputs in. The people that are polycropping are reducing their inputs hugely. I don't know how many of you are familiar with Derek Axton at Minton in Saskatchewan. He spoke just the other day at a workshop in South Dakota. And Derek farms 6,000 acres, and everything is pretty much polycrop now. He's got something like 30 or 40 different combinations of things that he's put together. He plants his crops together and he harvests them together, and he's got an optical grader, so he separates the seed after he's harvested it, and he is easily doubling his profit by planting two things together. He's cut back his synthetic ferts by 80%, and so he's using diversity to replace fertiliser. His soil is building unbelievably, and he had photographs of some new land that they've just bought next door, and he showed photos of that soil compared to his just across the fence, which goes, you could just see how much change they've made in the time that they've built their been building their soil. So I have no doubt that this multi-species everything is going to be the future. And there's certainly quite a lot of research going on into what things to put with what. Flax is actually a good thing to put with almost anything because it's not competitive at all. This was in Ontario, in Canada, where there was a demonstration farm, university research farm with a whole lot of cover crops grown as just monocrops, and then 3-way mixes, 5-way mixes, 7-way mixes, 9-way mixes, looking at a whole range of different things. But this was radish that was just grown on its own. It had actually all been fertilised, and right next to it it was obviously very nitrogen deficient, even though it had been fertilised. Right next to it was a mix of radish with oats and sunflowers and phacelia, no legumes in there. And the radish was completely completely fine in terms of nitrogen. There was no evidence of any nitrogen deficiency. Right next to each other, same amount of fertiliser on them all. So even putting non-legumes in there but increasing the diversity has solved the issue with the nitrogen. And that's just splicing those two photos and showing them side by side. And of course, Native Americans were very aware of the importance of diversity because they never grew corn on its own. It was always the three sisters, the corn, the beans and the squash grown together. So when the Spaniards talked about, the Spanish armies talked about marching up through Florida on one occasion where they were talking about cornfields that were 1 mile wide and 5 miles long, you think that that was all grown without fertiliser, without mechanisation of any kind, and huge amounts of corn being produced, corn grain being produced, using the three sisters. And in a part of the world now, if you go to those very same places in Florida, you can go to the very same towns that were exactly where the Spanish were talking about, where they'd been camping and finding all this grain which they plundered. But nothing will grow there now other than pine trees. It's just grey, gutless sand that seems to be totally infertile and considered by us clever people in this day and age to be incapable of growing corn or any other crop. So it just goes to show you that what you need is the life in the soil and the microbial activity and the diversity of plants, that even that grey gutless sand can be productive. So we hear a lot about the social network of trees and how trees look after each other. I particularly heard this when I was up in the northwest. And you'll see lots of stuff in the literature, like just to give you an example. This is an example of Douglas fir tree and a paper birch. So in wintertime, the paper birch doesn't have any leaves. And if you measure carbon flow in the soil, you'll see that carbon moves from the Douglas fir into the silver birch and keeps it alive over winter when it's not photosynthesising itself. In summertime, when the paper birch has got big leaves on it and it's photosynthesising much faster than the Douglas fir, and it's also shading out any little seedlings, tree seedlings that are in the forest, you'll see that carbon is moving from the paper birch into the seedlings of Douglas fir, for example, that might be being shaded by the paper birch. And the ecologists who've been looking at this go, oh, isn't this so lovely? These trees, they all care about each other and they're looking after each other and they're making sure that, you know, this one's kept alive in summer and this one's kept, and they're looking after each other's babies. Well, what a lot of bunkum. Who is actually looking after who there? Wouldn't it be the common mycorrhizal network? Wouldn't it be these highly intelligent microbes that are living underground that are going, well, this tree photosynthesises a lot in summer and produces lots and lots of sugars for us down underground. So we're going to keep it alive in winter we're going to take some sugar from the Douglas fir and transfer it across and make sure that we keep that paper birch alive because it's going to look after us. So what we see in actual fact is in multi-species mixes, whether it's trees or whether it's your cover crop, that it's the microbes under the ground that are responsible for diversity having such a big impact, because it is to the advantage of the microbes to have lots of different kinds of plants there. And it's to the advantage of the microbes to have plants that can photosynthesize at different times of the day or different times of the year, or respond to temperature differently or respond to rain differently. The intelligence is actually coming from the microbial community. All we actually have to do is make sure we have the diversity and let the microbes figure it out, because they're probably a lot smarter than we are. So one of the other things that happens here in the rhizosphere is that some of the microbes that live around plant roots move into the plant roots and decide that they're actually going to live in the plant. And all the other thing that happens, so that's what I was showing you that photo for, is that the plant will cultivate those microbes living there and then consume them. But it doesn't kill them, it just strips them of their cell walls, takes all the nutrients out of them, pops them back out into the soil again through the root hairs, and the microbes reform their cell walls, load up with nutrients again, and come back to the plant. So it's called the rhizophagy cycle or microbivory. And it's becoming you'll see more and more articles about this in the scientific literature. So it's a cycle. It's not just a one-way plants eating microbes. The microbes actually alternate between an intracellular endophytic phase, in other words, intracellular, inside the cells, inside the plant, and a free-living soil phase. So they can live out in the soil. So when you look at that photograph there, that's a new root tip forming on a plant, and what looks like a swarm of flies around it is microbes that have been attracted to that root tip because there's lots of exudates coming out. So the plant is sending out signals for the microbes to be attracted, but it's also signalling for exactly the kind of microbes that it needs. If it needs nitrogen, it'll be signalling for lots of nitrogen-fixing bacteria. If it needs phosphorus, it'll signal to those. If it needs zinc, if it needs copper, cobalt, boron, whatever it is that it needs, it's going to signal to those microbes and feed those microbes. It's going to attract them to the root tip. So remember, these are newly emerging roots. There's another reason why you want your plants to be young, vigorously growing. Look at your grazing management, make sure you've got new roots forming all the time, because this is where all of this activity is going to happen. When the microbes get close enough to the edge of the root, it's actually engulfed by the root, takes them in. It's called endocytosis, pulls them in. As I said, they get subjected to reactive oxygen, get their cell walls stripped off, all the nutrients are taken out, but the microbe doesn't die. The microbe might even multiply at that stage, moves around in the plant, and you'll find lots of stuff about this on the internet. In fact, there's a really good podcast. John Kempf did an interview with James White a couple of weeks ago. You'll find that on the net, and also a really good YouTube video that's got lots and lots of photos of what this actually looks like inside the plant. But in short, basically plants nurture microbes in the root zone. They enter the root tip via the soft meristem cells, so this can only happen in young roots. Microbes are exposed to reactive oxygen and breaks down their cell walls, releases the nutrients. The internalised microbes stimulate the formation of root hairs, which is why you will see far more root hairs on plants that are interacting with microbes, and you'll see very few root hairs in situations where you're using lots of chemicals. And then they will exit the plant via the root hairs and start the cycle all over again. So this brings me to the endosphere, which is very close to the end of this talk. Endo means inside. So the endosphere is what happens inside a plant. It's the microbes that live in plants, and we call them endophytes. So endo means inside and phyte means plant. So anything that lives inside a plant is an endophyte. But what's very interesting about endophytes is that they can be microbes that were just living in the soil, and they got invited in, or they decided they wanted to go in and live with the plant, and they will live with the plant for its entire life. And sometimes they'll end up in the seed, so that when that plant forms seed, the microbes that it took from the soil earlier end up in the seed, and when that seed germinates, those microbes help the newly establishing plant. They help it find nutrients, they fight off pathogens, they protect it from stresses like drought or frost. So they're very, very protective of that seed, and then that plant will be more vigorous than its parent plant. And then it will take up more microbes from the soil and take them through to the seed. So if you're growing plants in biologically active soil, where the soil is building and you keep on keeping the seed keeps on getting better. And many people are noticing this. So the seed will germinate with more vigour. It will have more resilience for pests and diseases and also abiotic stresses. Now, when I was talking to Derek Axton about this the other day, who's the guy I mentioned who's polycropping, and he's cut back right on his synthetics, and he puts vermi-liquid on the seed, and he's doing all kinds of really great things, making heaps of money, which is great. He said that if they buy a new variety of something they haven't planted before, they'll buy certified seed, and they notice the first year they plant the certified seed that it is nowhere near as vigorous as anything else that they're growing on their farm, because it's come from a situation where it's been grown with lots of fertiliser and chemicals and stuff like that. They'll then keep that seed, and it'll have a much better core microbiome. I haven't mentioned the core microbiome yet, but the core microbiome is all the internalised microbes, And then they plant that and it grows more vigorously, and then they keep that seed and the next year it grows more vigorously, and they're noticing that whenever they bring new seed in from somewhere else, even though it's certified seed, because it's grown in soil that's not biologically active, it's nowhere near as vigorous as their own seed. So people are noticing this. It's a very real thing. It has very practical implications. On the other hand, if you're growing plants in soil that's not biologically active, the plant will not internalise many microbes from the soil. It won't have many endophytes in it. It won't be able to get its own nutrients. It won't be able to defend itself from pests and diseases, and it won't internalise many microbes in the seed. So even if you keep that seed, it's not going to be very vigorous. So when we're talking about the endosphere, there's two things to consider. One is the core microbiome. You'll find lots of information about that on the internet, which is basically the microbes that a plant is born with, if you like. In the same way that we humans are. We get microbes from our mothers when we're born, and the microbes are in a plant, basically they're maternal. They're coming from the mother plant. So one thing is what microbes is a seed, what does a seed have in it, what is its core microbiome, and the other thing is biological induction, like how many microbes can it get from the soil. So the healthier that you can get your soils, the more biological induction you can get, and the better the core microbiome will get if you keep your own seed. So the core microbiome, it's species specific, it's cultivar specific. It's an assembly of microbes that basically just belongs to that type of plant, and it's not just bacteria, it's protists and archaea and fungi as well, because they move out into the and then after they've surrounded the seed when it's germinating from the rhizosphere, they then move back into the plant, into the stems, into the leaves, into the flowers, and into the seeds, and begin the cycle all over again, and remains within plants throughout their life. So I think I've already told you all that. So biological induction is just free-living microbes, all kinds of amazing things that you would never think would move out of the soil into plants will do so if it suits them to move in there, and they may stay there for the rest of the plant's life. They're very significant. They're very significant for things like biological nitrogen fixing. So, for example, I mentioned the Haggertys. The Haggertys are in your book. I think they're on page 32 or something. I was looking at it the other day. If you take a soil test, if you take a soil sample from their farm, send it off to the lab, come back saying there is no nitrogen in this soil. So how can they possibly grow a wheat crop? When you send their leaf samples off to get a tissue test or a leaf test or whatever you call it in the United States, it says there is optimal nitrogen in the leaves of this plant. It's right in the sweet spot, exactly where nitrogen should be. But there is none available in their soil. When they get their grain tested at the elevator, the guy doing the test, if he doesn't know them, said, "Where did you get this grain from? You didn't grow this in Western Australia. You didn't grow this on sand. Your protein level is higher than any other wheat grown in this region." So how can they have the highest protein content of any wheat in the region, absolutely optimal levels in their leaves and none in their soil? Because you don't want to have any free nitrate in your soil. That's not where you want it to be. You want to have lots of microbes that are living in your plants and fixing nitrogen from the atmosphere, even in wheat plants. Doesn't matter what kind of plant it is. So that's what's going on their farm, is that they have all these endophytes. So you need to look at, well, how are they managing their seeds and how are they managing the way they grow their wheat in order to have a biologically active system. So I'm sure Marlon talked about the 5 principles for soil health this morning. So I'm not going to ask him what they were. Can someone tell me what they are? I'm sure he would have mentioned it, yes? Living roots in the soil, yep. Armour on the soil. Animal integration, biodiversity, that's 4. Minimise disturbance, 5. Okay, so they were our 5 soil health principles. What do you think those 5 soil health principles do for microbes in the soil? We're going to minimise soil disturbance. That's going to be good for microbes, isn't it? We're going to maximise soil cover. That's going to make it much more comfortable for everything that lives in the soil. Plant diversity, we know how important that is. We're going to talk about that a little bit more just in a minute. Continual living plant roots, yes. Keep microbes alive, and livestock integration, very, very important. Lots of signals come from livestock. Manure, urine, are absolutely full of biochemical signals, act as biostimulants to microbes in the soil. Go, wow, this is a great place. You know, there's animal integration. It's more living, it's more vibrant, it's more wholesome, if you like, for when you're looking at it. For all these things that are living in the soil, the only thing they can respond to are biochemical signals. And if you have animals integrated, there's a lot more biochemical signals as well. So if we just look at the plant diversity aspect of that, because I figure that that's the thing that people have really cottoned on to, like with multi-species covers, and now we're talking about, like I said, if you look in your book, relay cropping, interseeding, polycropping, all kinds of ways of cropping as well. And just looking at it from an animal, like just looking at the forages, we know it improves animal nutrition hugely, especially if you can get those Brix levels up. We are seeing in places like New Zealand where there's everything is out on pasture, really. We don't have any confinement feeding in Australia or New Zealand. Improves growth rates of animals, improves milk production for the dairies that are grass-fed dairies, pasture-based dairies, improves conception rates in their cows quite dramatically, reduces dependence on vets don't like, and builds soil. So we're seeing all these advantages through plant diversity, but these diverse systems are self-organising. That's the key thing, that the microbes actually know what to do. So all we really have to do is manage for above and below ground diversity, and the details will take care of themselves. And we do need more flowers. This is just my final parting comment. As I mentioned earlier this morning, the prairies had hundreds of different species of flowers in them. So we really need to think about, take this seriously, getting more flowers back into systems. There's so many flowers that animals will eat and we can eat as well. Having gourmet flowers is becoming a thing in some of the top restaurants. They'll put flowers on your plate as well as the food. I'm not sure whether this would grow here. This is sheep's burnet. It's very, very drought tolerant, and it's also cold tolerant. Is anyone aware of that plant? I'm not sure. Does it look like that? Yes. So maybe it might be the same thing, just with a different name. Yeah, that's one of my favourites for inclusion in pastures. Also, it's extremely high in secondary plant compounds, which are very important for animal nutrition, feed conversion efficiency. Some of these things that may not be the bulkiest plants on the planet really improve feed conversion efficiency, so you don't need as much dry matter to get good live weight gains. Chicory, of course, in the daisy family. It seems to be a universal wherever I go in the world. People love chicory. Evening primrose is another one. Not sure whether it would grow here, but it's incredibly drought tolerant and very high in secondary plant compounds. Yarrow. I saw yarrow. Oops. Evening primrose grows here. Great. I saw yarrow growing over in Oregon, so whether or not it grows here, I'm not sure. This one's tansy, which is not the same thing as ragwort, but it's in the same family. Cosmos, one of the favourites. There was a yellow cosmos that was a native plant here. Well, I'm sure it's still here. It was a native plant. Was the leaves and the flowers were eaten by Native Americans. Very, very nutritious plant. Cows just love cosmos. This pink one is a native of South Africa. You'll only ever see it growing on the side of the road because cows love it so much that, and it's one we can eat. We can eat the petals of this as well. But if you've got good rotational grazing, you'll be able to get cosmos growing in your paddocks. That's a delphinium that's just growing in my vegetable garden at home. There's lots of different vegetables all mixed in together. We always see insects on these kinds of things. That's just cosmos and sunflower. corn and celery, like all growing together, just in your vegetable gardens at home, put all the things in together, just like what you would in a cover crop. This is a cane farmer this is sugar cane in Queensland. All of our sugar comes from cane, and he's planting sunflowers in between the rows of cane because cane takes quite a while to grow. So he's seen huge benefits in his cane from integrating sunflowers in. You can see a little bit of sugar cane just at the front there. Again, in vineyards and other horticultural systems, think about your ground cover and think about getting flowers in there. This is the STRIPS program. This is US. This is corn. I think you can have up to 10% of the area of corn with prairie flowers in the strips and still get the same yield, but see huge advantages in terms of not needing insecticide because of the predatory insects that come and live in the strips. And also farmers that have gone in for the strips in corn have talked about the big numbers of increases in birds that they're seeing on their farms. So it's just called STRIPS, strips, and they're prairie strips in corn. There's green cover seeds, which are the people that have put together the booklet that you have. They have lots of field days there in the summertime. That's Keith Burns, just looking at different kinds of mixes of flowers and things that you can have. That's flax or Phacelia. I can't see from here. Phacelia, sorry. Yeah, so think about the flowers. And the thing about integrating lots of different things is that there are some plants like, say, flax, for example, that's not a high biomass plant. It's not competitive with anything you can put flax in and it won't detract from the yield. It will probably increase it. It's highly mycorrhizal. So when we're thinking about what to put together in our cover crops, they don't always have to be high biomass things like sunflowers, for example. Yes, put some of your plants with your big leaves, put them in, but remember as much diversity as you can as well. So I'm out of time, so I'm going to end there before I get dragged from the stage, but I just got to the end just right at the perfect time. So thank you very much for listening.